All right, so this is the second of two videos regarding metabolism. And where we ended last was we had a big fat handful of NADH and FADH2. These are the molecules that are carrying the electrons that we had stripped from glucose. And now we're going to take them to the electron transport chain where they will set off a series of redox reactions, so reduction oxidation reactions, that because they are exergonic will release energy, and then that energy is going to be used to do work. So let's take a look at how this all plays out. So here we have our three phases of glucose metabolism. We've looked at glycolysis here within the cytosol. We saw the pyruvate decarboxylation that occurs here in the matrix. And then we saw what happens when those two acetyl-CoAs are completely oxidized to CO2 here in the citric acid cycle. And so now we've collected the NADH from glycolysis, we've collected the NADH and FADH2 from the citric acid cycle, and the NADH that had come from the pyruvate dehydrogenase complex, not shown here. And we're going to take them over here to the inner membrane of the mitochondria where oxidative phosphorylation occurs. And oxidative phosphorylation is a combination of the electron transport chain and chemiosmosis. And it's there that the bulk of ATP will be made from glucose. So let's have a look. The electron transport chain is located in the inner membrane of the mitochondria. So if we take a look at a mitochondria, we have an outer membrane, and then we have an inner membrane. And now that inner membrane has all these folds. Those are called cristae. And the reason they're there is because they provide that inner membrane with more surface area. And the more surface area that is, the more of these proteins that membrane can host. And then inside, so that new compartment inside there is called the matrix. And the compartment that exists between the inner membrane and the outer membrane is called the intermembrane space. The proteins that we're going to be interested in are all transmembrane proteins that span the inner membrane. And what we're going to see is as the electron transport chain functions, as these redox reactions are occurring, the energy that's being released is used to pump protons from the matrix into the intermembrane space. So let's take a detailed look at this. This figure will give you everything you need to know about the electron transport chain. And so let's just familiarize ourselves where we are. So right here, this is the inner membrane of the mitochondria, which means out here in the orange, this is the inter, with a T, intermembrane space. And here is the matrix. Here's that NADH that came from the citric acid cycle. Here's the FADH2 that came from the citric acid cycle. They're carrying the electrons that ultimately came from glucose. And the most important points here is complex 1, complex 2, complex 3, and complex 4. Each one of these complexes are transmembrane enzymes. They are enzymes that catalyze redox reactions, meaning they catalyze the transfer of electrons from one molecule to another. And you need to know which molecule becomes oxidized and which molecule becomes reduced in each one of these enzymes. You should be able to kind of tease it out on your own, but let me help you out with that. In complex 1, so complex 1 is an enzyme that catalyzes the oxidation of NADH. So we're removing the electrons from NADH and we're giving them to Q. Now Q is called ubiquinone, and it is hydrophobic and therefore lives within the inner membrane itself. And it is a reactant and a product, right? It is a product in that it has just become reduced. It is a reactant for the next reaction. So not only is it going to steal electrons from NADH, okay, and that's catalyzed by 1, but it is also going to give electrons to cytochrome C, which happens here in cytochrome 3. And so Q has the ability of moving around the membrane, going to complex 1 and then going to complex 3. In addition, it also goes to complex 2. Complex 2 is another enzyme that catalyzes the oxidation of FADH2 and the reduction of Q. In other words, complex 2 catalyzes the transfer of electrons from FADH2 to Q. Okay, so there's two ways that we can reduce Q. Through complex 1 and NADH, and through complex 2 and FADH2. Now, Q is now reduced, and it goes over to complex 3. And complex 3 is going to catalyze the transfer of electrons from Q to cytochrome C. Now, cytochrome C is another electron. It's another electron carrier, but this electron carrier is not hydrophobic. It's hydrophilic, and so it exists out here at the surface of the membrane, and it accepts electrons from Q. It is catalyzed, right? That reaction, that redox reaction, is catalyzed by complex 3, and then finally cytochrome C. Now it's reduced. Now it has these electrons. It passes those electrons to oxygen with a couple protons to become water. And that's catalyzed by complex 4. Now, each one of these reactions are they are an exergonic redox reaction, which means they release energy. Now, complex 1, complex 3, and complex 4 couple that energy release, couple that redox reaction with the endergonic pumping of protons from the matrix to the intermembrane space, right? This is coupling. This is energy coupling. We are using an exergonic reaction to couple to an endergonic reaction to get that endergonic reaction to become exergonic, to actually occur. So by the end of this, right, oxygen is the last electron carrier. It has the highest affinity to electrons. That's how this entire electron transport chain works. That oxygen has a higher affinity or higher electronegativity than cytochrome C. That's why that reaction is exergonic. Cytochrome C has a higher affinity to electrons than Q. That's why that reaction is exergonic. And Q has a higher affinity to electrons than either FADH2 or NADH. That's why that reaction is exergonic. Right? So we have increasing electronegativity as we reach oxygen, and oxygen has the highest electronegativity within even all of the animal cells, right? It's why this whole thing works. It then becomes H2O and it steals those electrons, and that H2O then is excreted, and then you breathe in more oxygen and you can get this process occurring over and over and over again. Okay, so now what we need to do is figure out where is the bulk of energy from glucose. We have a couple ATP from substrate level phosphorylation that happened in glycolysis. We have a couple ATP that happened via substrate level phosphorylation in the citric acid cycle. But that's not the bulk of the energy that came from glucose. Where is it now? And the answer is this concentration gradient of protons. We call that concentration gradient, we call that potential energy of water behind a dam, we call that the proton motive force. And this is where the bulk of glucose's energy now exists, and this is the energy that we're going to use to do the work of making ATP. So how does that work? First, before we go into that, I just want to stress that each one of these complexes is an enzyme that catalyzes a reaction. And even as an enzyme, they have enzyme names. Complex 1 is actually called NADH-Q oxidoreductase. And here's the reaction that it catalyzes. You do not need to memorize all of this. I just want you to see that these complexes are simply enzymes that catalyze redox reactions. That's it. Okay, so how does the cell harness the energy of the proton motive force to make ATP? Well, that process is called chemiosmosis. So it's the second half of oxidative phosphorylation. And it uses this proton motive force here within the intermembrane space. As those protons move down their concentration gradient, that is how ATP is going to be made. And the enzyme that does that, the enzyme that harnesses the exergonic flow of protons down their concentration gradient to the endergonic production of ATP from ADP and inorganic phosphate is ATP synthase, and it looks like this. So here we have our intermembrane space. This is where all those this is where the proton gradient is. And what happens is as those protons move through, it provides this enzyme provides a channel for those protons to move through. Otherwise, they can't get through that inner membrane. In fact, it's absolutely essential that they cannot get through that inner membrane any other way than going through this passage of ATP synthase. And as they pass through, it quite literally spins this catalytic, this rotor here, which then interacts with this catalytic knob, and that is the energy necessary to take this, what is normally an endergonic reaction, ADP plus inorganic phosphate to make ATP, and make it into an exergonic reaction, coupling the proton flow to making ATP. So, if we were to experimentally figure out just how much ATP can be produced from the energy of NADH, from the number of protons that NADH fuels to be pumped out, and how much FADH2 contributes, what you would find is this, is NADH per NADH, we get 2.5 ATP made. Per FADH2, we get 1.5 ATP made. Why is there a difference? Well, let's take a look. FADH2 passes its electrons to Q from complex 2, therefore bypassing complex 1. So electrons gained from FADH2 bypass this, meaning that it doesn't contribute to any protons being pumped out by complex 1, whereas NADH starts right here at complex 1. And so we get the protons pumped from complex 1, 3, and 4. Whereas from FADH2 only contributes to the pumping from complex 3 and complex 4. All right, now we have all of the tools to calculate the total amount of ATP necessary, or the total amount of ATP produced from glucose. Here it is. Glycolysis produced 2 ATP via substrate level phosphorylation. The citric acid cycle produced 2 ATP from substrate level phosphorylation. And then, right here, for all of the NADH and the FADH2 that comes from glycolysis, that comes from the pyruvate decarboxylation, that comes from the citric acid cycle, when we multiply it by 1.5 or 2.5, we get 26 or 28, which then comes out to either 30 or 32. And notice we have or both here and here. Why or? Why or? Here's the reason why we have an or there. The reason is it depends upon what cell type we're talking about. Because there is 2 NADH here in the cytoplasm that needs to get into the mitochondria. The problem is NADH can't get through the inner membrane. So there is an indirect shuttling mechanism to get those electrons from the cytoplasm into the matrix. Some cell types use a shuttle in which those electrons from these NADH are shuttled to electrons in these NADH here within the matrix. In that case, no energy is lost and we get the full 32 ATP. However, in some cells, a different shuttling mechanism is used. The electrons in these two NADH are shuttled to electrons here in FADH2, which means even though we're supposed to get 2.5 ATP from each of these NADHs, we're only getting 1.5 ATP from each of them, which means that we're losing a couple ATP. So in those particular cell types, we're only getting 30 ATP per glucose. In the next video, we are going to talk about fermentation. So now let's talk about fermentation. Okay, fermentation is kind of like one or two extra reactions that occur after glycolysis in anaerobic conditions. And fermentation occurs in some of our cells. Fermentation happens in lots and lots of microorganisms. In fact, yeast, right? Yeast, we use yeast because they perform fermentation pretty often, and the CO2 that comes out of yeast fermentation allows bread to rise. The ethanol that comes out from yeast fermentation gives us alcohol for beer. Bacteria have lactic acid fermentation. The lactic acid they produce leads to yogurt. The lactic acid those bacteria produce leads to sauerkraut or kimchi. So let's take a look at what is fermentation on a biological level. Well, it solves a very important problem, and that is this, that there are sometimes, well, there's some cells that exist that don't require oxygen at all, or don't use oxygen at all, and there's sometimes in our cells that they need energy and they're not getting enough oxygen to supply it. When that's the case, oxidative phosphorylation shuts down without oxygen. The citric acid cycle shuts down without oxygen. The only thing that is not dependent on oxygen is glycolysis and those 2 ATP that are produced per glucose that's converted to 2-pyruvate. But even glycolysis in the absence of oxygen would shut down for this problem here. That as glycolysis occurs, it uses up ADP, inorganic phosphate, and NAD+. However, that ATP that's made is recycled right back into ADP and inorganic phosphate when the cell uses the ATP. And that NADH that's produced goes straight to the electron transport chain, dumps its electrons off over there, and recycles itself as NAD+ to be used over and over and over again. And so that's great. But in the absence of oxygen, that means that NADH can no longer dump its electrons off at the electron transport chain. And so that means it's just building up, building up, building up, building up. We're running out of NAD+, running out of NAD+, running out of NAD+, and eventually even glycolysis would discontinue, would stop in the absence of oxygen. But fermentation provides an alternative pathway to recycle NAD+ in the absence of oxygen. And there's two types of fermentation, two sets of reactions that allow cells to do this, to oxidize NADH in the absence of oxygen. The first is lactic acid fermentation. And the way it works is this. Here's glycolysis, glucose to pyruvate. Here's the NADH being produced, and here's the 2 ATP that are being produced. Well, that NADH is going to be building up if we don't send it to the electron transport chain. So in the absence of oxygen, what the cell does is it sends its electrons to pyruvate. So pyruvate becomes the oxidizing agent, the thing that steals electrons from NADH, recycling it back to NAD+. When pyruvate does that, when it steals those electrons from NADH, it becomes lactate or lactic acid. And so that is how the cell, that is the one reaction of fermentation. And again, its purpose is to use some alternative method of oxidizing NADH back into NAD+. And in this particular case, pyruvate is that molecule to do that, because otherwise pyruvate can't be sent to the matrix, because without oxygen, the citric acid cycle doesn't work. So it's just kind of sitting around doing nothing anyway. Now, ethanol fermentation occurs in things like yeast. Lactic acid fermentation occurs in our cells. Ethanol fermentation occurs in cells like yeast and some bacteria. And it is now just two extra reactions. So here's glycolysis, here's the formation of NADH, here's the 2-ATP that's giving the energy for those cells to survive. Even the cells who never use oxygen, they get plenty of energy from this 2-ATP that's produced per glucose. And then here's our 2-pyruvate just sitting around because it can't go to the matrix because there's no oxygen. So what happens? Well, there's one reaction that first decarboxylates pyruvate into acetaldehyde. So boom, off go to CO2. And then it's acetaldehyde that is going to oxidize NADH. Not pyruvate, but pyruvate's derivative, acetaldehyde. And so that recycles NADH into NAD+, and the byproduct of acetaldehyde, when it steals those electrons, it becomes ethanol. There is your alcohol in your beer, in your wine, and so on and so forth. Make sure, I've been using all of these different phrases, but they all mean the same thing. Pyruvate oxidizes NADH. Pyruvate steals electrons from NADH. NADH reduces pyruvate. NADH gives electrons to pyruvate. Each one of these four phrases means the exact same thing. So make sure you understand that. All right, almost done. Now, it may seem as though glucose is the only thing a cell can metabolize, and that's obviously incorrect. That there are plenty of other reduced fuel sources that can be used in metabolism, that can be oxidized. Fats can be broken down into glycerol and fatty acids. Proteins can be broken down into their amino acids. And then there's another set of reactions that will convert glycerol into an intermediate of glycolysis, that can convert fatty acids into acetyl-CoA, that can convert amino acids into pyruvate, acetyl-CoA, intermediates of the citric acid cycle. So basically, what you have is a situation where these reduced fuel sources are, through various enzymatic means, broken down into some intermediate here, some intermediate of glycolysis, acetyl-CoA, or the citric acid cycle. And then once they reach that intermediate, they're in familiar territory, and you know what happens from then. So this is then how your cells can metabolize these other reduced fuel sources. Finally, these metabolic reactions are regulated. There are times when the cell has enough energy, plenty of energy. So why break down glucose when you got plenty of energy? Or there's times when the cell doesn't have enough energy, and it needs right to tell these reactions, these enzymes, to okay, start breaking this stuff down, start breaking this stuff down. And there are just a few enzymes in each of these pathways that are regulated, that act as allosteric enzymes, that are allosteric enzymes. So you can think of this intuitively. What signals to the cell that, hey, plenty of energy here? ATP. So ATP is oftentimes an allosteric inhibitor of many of these regulatory enzymes. So let's take a look at phosphofructokinase 1. This is another enzyme. Put on your list. You're going to need to know it. It's an enzyme involved in glycolysis, and it is one of the three enzymes of glycolysis that is regulated. It's an allosteric enzyme. And there are two regulators that turn it off. ATP turns it off, and citrate, which is a downstream product. If you got lots of citrate, the cell says, we're good, we don't need any energy. It binds to phosphofructokinase and turns it off. Now, AMP is signaling to the cell, uh-oh, we've been dephosphorylating all of our ATPs. We've got too much AMP here. We need energy. It binds to a regulatory site on phosphofructokinase, activates it and gets glycolysis running again.